Bismillahirrahmanirrahim. Assalamu alaikum. How are you, dear students? I welcome you in Pakistan International School TAIF's virtual learning system for the session 2020, and this is biology class for first year. Dear students, today we will see about photosynthesis, and as you know that we have started chapter number 4, bioenergetics, and in that we are discussing photosynthesis. We have seen some of the important points like about pigments, chlorophylls, importance of water and carbon dioxide and light for the process of photosynthesis. We know that photosynthesis is divided into two reactions, light reaction and dark reaction. So today we will study about light reaction. So let's get started. Dear students, if we see that mechanism of photosynthesis, how this process is going on. So you know that with the help of water and carbon dioxide and plus water in the presence of chlorophyll and sunlight, we get what? Carbohydrate, that is in the form of glucose, and then we get here very important gas, oxygen, and we are getting H2O, means water as well. So this all process of photosynthesis just taken out in two steps. First is light reaction. It means that this is light dependent reaction in which the light process, the light of sunlight is used here. So the other one is dark reaction, which we'll see in the next lecture, inshallah, which is light independent reaction. It doesn't mean that light independent reaction is just done in the night time. No, it is done in the daytime, in the light time, but light is not needed for that reaction. That's why that is called dark reaction. So the products which are formed in the light reaction, they are used in the dark reaction. So light reaction is said to be the first process of photosynthesis, and the most important event of light reaction is the production of ATP, and the production of ATP, that is by the process of phosphorylation. Phosphorylation means we add phosphorus or phosphate group with adenosine diphosphate. So it will become adenosine triphosphate by using sunlight, and this is called photophosphorylation. It means by using sunlight we are doing phosphorylation. In phosphorylation or photophosphorylation, light energy is used to create a high energy electron donor and a low energy electron acceptor. So electrons then move spontaneously from donor to acceptor through an electron transport chain that is also called ETC, electron transport chain. So in electron transport chain, via moving electrons from donor to acceptor, ATP is formed. So photophosphorylation, that is are of two types: cyclic photophosphorylation and non-cyclic photophosphorylation. So here we will see cyclic photophosphorylation. So you can see here light reaction and dark reaction. In this, light energy is used. So we are going to talk about this step here. One by one. So there is photophosphorylation in which ATP is synthesized, and then ADPH is also synthesized, which are highly energy compounds which are used in Calvin cycle to give the plants sugar molecules or carbohydrates. So in this light reaction, first step is a cyclic photophosphorylation and non-cyclic also. We will discuss what is the difference and why they are how they are how they form that ATP. So we'll see non-cyclic photophosphorylation. In non-cyclic photophosphorylation, solar energy strikes the antenna complex of PS2. So we know that this is complex PS2 and this one is complex PS1. This one is discovered first and PS2 discovered second, but the reaction is going on from the second to one. So this is 680 nanometer. It absorbs maximum and it absorbs maximum wavelength of 700 nanometer. So what happened? The PS2, that is around it, there are lots of molecules present over it, and these molecules are different pigments, including carotenoids and chlorophyll B. So light strikes over it, and electrons get excited from every molecule, and then that energy from that electron is transferred to reaction center, which is chlorophyll A. So electron from this chlorophyll molecule absorbs this energy and become excited. So what happened from here? The electron gets all the energy from this system and gets excited and leave its place. So excited electron is then caught by primary electron acceptor, which is above it. Like any molecule, we'll discuss it in detail, and then higher energy level. While the water undergoes photolysis to replace the just excited electrons from the oxidized P680 chlorophyll. This also generates 2H ions and O2, which is lost through the stomata. So here photolysis is going on, and when the water is broken down with the help of in the presence of sunlight, then these electrons are passed on through electron transport chain like this, and they move through PS1, and which reduces the energy of electrons and dissipate in amount that is enough to produce ATP carrier molecules via ATPase machinery, and then first product of light dependent stage. So here the first product is T and P, high energy molecule, which is going to be formed during electron transport chain. So how this whole process is done, we will see here. And why it is said to be non-cyclic, because the electrons which are moving on after passing through PS1, it just change into NADPH2. It does not like the products, and those will not come again into the photosystems. That's why they are called a non-cyclic photophosphorylation. So we will take this step by step. So first step of this reaction is absorption of light by PS2 and excitation of electrons. So as we have discussed before, this is photons. So when just example, 2 photons just fall on these antenna molecules of chlorophyll and carotenoids, all the pigments which are accessory pigments here. So light is absorbed by every pigment, and this pigment and the energy like by which these electrons are excited, that energy just collectively reaches to this P680, and P680 is the reaction center in which chlorophyll A is present. So this is photosystem 2. So these 2 photons just removes 2 electrons from here. After excitement, it just goes. So absorption of light by photosystem 2 and excitation of its electrons. Now electrons are excited here, and they are captured by primary electron acceptor. That primary electron acceptor is plastoquinone. Okay, the second step of this one is photolysis. The same time when the electrons are emitting from PS2, at the same time, by the help of this light energy as well as the chlorophylls B and A. So what happened? This water is broken down into, or it's split into two protons and oxygen atom as well as two electrons. So by this, this is with the help of water splitting enzyme, because we know there are lots of enzymes which are going on during the reactions, metabolic activities, and especially photosynthesis and respiration. So this indicates as Z. So by this by this water splitting enzyme Z, just example, just given the name Z. So water is broken down into two electrons, two protons, and one oxygen atom. So by this, in this step, very, very important step is release of oxygen in the form of molecule. So four water molecules are broken down, and four hydrogen, this proton will be formed, and oxygen molecule will be formed in that form, and then it will be released out of this stomata. So there is overall a reaction of this one, that water by the photolysis, it is broken down into or split water into two proton, two electrons, and the water molecule. Then come to the next step of this. Now electron flow from PS2 to PS1. So this PS2 here, when electrons get excited, so there must be an electron acceptor which accept this energetic molecule, energetic electrons. So these electrons are just get into, or they are accepted by a plastoquinone. So that is primary electron acceptor, and that is iron containing protein. So this is iron containing protein. So from PQ, electron flow through the cluster of another iron containing protein, that is a cytochrome. Cytochrome complex, we can say that, because cytochrome B6, cytochrome F, that is a cytochrome complex. So these molecules can just present in this electron transport chain. So during this electron transport chain, what happened? The electron flow through the cytochrome complex. When it flows through here, it stimulates it to pump the protons from stroma to thylakoid inner surface. That is inside, we can say, the chloroplast. Yes, this you can say this is chloroplast, or a thylakoid. Yes, a thylakoid. So in the membrane, we have seen before in the previous lectures that these photosystems are present. So this is outside of this thylakoid membrane. So this is a stroma, and now this proton just pumped out from stroma into the thylakoid membrane inside of that inner space. So this is very, very important, because from outside, gradient of proton is shifted towards inside of the thylakoid membrane. So means here proton gradient, now it rises up. So this is the point that chemoosmosis is going to be done. Chemoosmosis means, now, after this, there is the next step, osmotic synthesis of ATP. Now, this proton gradient is inside this inner membrane space of thylakoid, so proton gradient is higher here. How ATP is going to be synthesized, this is step we will see in the next diagram, where we see chemiosmotic synthesis of ATP. Okay, so when absorption of just continue the previous one, that ETC, or electron transport chain, is moving the electrons from higher energy level to lower energy level. So next electron acceptor is plastoquinone, and this is this is not plastoquinone, and this is plastocyanine. Okay, this plastocyanine, that is copper containing molecule. PQ is iron containing molecule. Now copper containing protein or copper containing molecule that finally gives this electron to PS1. Now absorption of light by PS1, excitation of its electrons. Now what happens here, the same process is going to be done as it is in PS2, but in PS1 there is no photolysis of water. So light, when strikes on PS2, PS1 at the same time it strikes over there, and by all the same method, the electrons get excited and reaches to reaction center of P700. P700 means it can absorb maximum wavelength up to 700. So then the two electrons get excited and move to the primary electron acceptor. Electron acceptor here is ferredoxin. Okay, then we are having the next step, electron flow from PS1 to NADP. Now what happened in the next step? The primary electron acceptor of photosystem 1 passed the photoexcited electron to the second electron transport chain. This is this one, where the electron acceptor is ferredoxin, and this is also iron containing protein, and an enzyme called NADP reductase that accept these electrons, these two electrons from here, and proton also from inside, and especially the protons which are released by the splitting of water, that moves here, and two protons from here, and NADP is present over there. So this NADP reductase enzymes is now going to be perform its function and accepting these two electrons. So that is reduced into NADPH. NADPH is a highly energetic compound which is later on utilized in Calvin cycle, or that is called dark reaction. So here we can see this its equation, NADP plus 2E, like 2 electrons plus 2 protons, so it will get NADPH plus H. So that is highly energetic molecule. Here the function of NADP reductase is very, very important, which is accepting these two electrons and give this product. Now what happened? Here the two electrons which are excited and just get out of this place. So here at this place, chlorophyll A, two electrons holes are formed. Holes means empty space, electron space. So what happened? The electrons which are formed by splitting of water here. So these two electrons quickly move towards this to fill that space. So there is no hole left over there, electron hole. Like that in photosystem 1, the electron holes are formed by excitement of these two electrons which move and accepted by ferredoxin. So these holes are then filled up by the electrons which are moving down from the electron transport chain of photosystem 2. So there are also no electrons. So they will get excited by just accepting this energy again, and then they just move towards electron acceptor. So this is non-cyclic procedure, or that is called Z scheme, because path of electron transport through the two photosystems during non-cyclic photophosphorylation is Z scheme due to conceptual zigzag, like this zigzag formation of this one, zigzag shape. So let's talk about chemiosmosis, chemiosmotic synthesis of ATP during light reaction, which is going to be done in this process, in this reaction where the electron is moving through the complex or B complex. Okay, so let's see, this is a chloroplast stroma, and from here this is thylakoid membrane. So while moving these electrons from P6T 680 to plastoquinone and then ferredoxin complex B6 and F, here electrons, this PQ and PQH2, these are also the complex plastoquinone, isomers, like the changing forms of this in a protein. So when electron passes, this energy is utilized to move down proton from stroma to the inside of thylakoid membrane. Here the oxygen is evolved. From here this oxygen is removed out as molecular oxygen. So one proton is formed from the splitting of water, and the others, such the rush of proton just gets inside through influx of proton through this plastoquinone, and when this electron moves through B complex. So this is going to be happen here. This one, actually this process is going to be done here with this one, because here proton gradient is more in the inner side, inner space of thylakoid membrane. So here is an enzyme that is called ATP synthase or synthetase. So what happened? It is going to perform its function, and it moves the proton outward now, because now outside the proton gradient is less. So from higher to less concentration, it just moves outward. During movement of proton from inner to outside in the stroma, this energy of movement of this proton, this energy is utilized to phosphorylize ADP with PI, inorganic phosphate. So when these two combine, they form ATP, and that is the desired product of photophosphorylation or light reaction. On the other hand, when the plastocyanin accepts and it gives 2 electrons to PS1, here also the excitement of electrons is done. So it just go excited and just move out, and the electron accepted by ferredoxin and FNR, ferredoxin NADP reductase. This reductase, NADP reductase, is an enzyme or protein molecule which reduce NADP and plus H, and with the help of these electrons, NADPH is formed. So these two are the main products of this cyclic reactions. Like these energy are utilized in the dark reaction. Let's talk about cyclic photophosphorylation. Cyclic photophosphorylation involves photosystem 1 only, and light is absorbed by photosystem 1 is passed to the primary pigment, and an electron in the chlorophyll molecule is excited to a higher energy level and emitted from chlorophyll molecule, that is called photoactivation. Like a same electron is getting excited and is moves to electron transport chain. So what happened in this reaction? Instead of falling back into the photosystem and losing its energy as thermal energy and as fluorescence, the excited electron is captured by electron acceptor, like here. Ferredoxin is there in this. So ferredoxin is there and passed back to the chlorophyll molecule via a chain of electron carriers. So here, what happened? Uh, these electrons does not produce NADP. NADP or does not for DPH, does not reduce NADPH. So what happened during this process? Enough energy is released to synthesize ATP from ADP and inorganic phosphate by the process of chemiosmosis. ATP then passes to the light independent reaction. So this is actually, if we see through this diagram, like if we talk about first procedure, photosystem 1, electrons get accepted by primary acceptor, plastoquinone, here ATP is formed, okay, like this. But sometimes it happens that ATP is deficit, and in deficient amount, it's not in enough amount, but NADPH is formed in much amount. So we don't need then this one NADPH, we need ATP, plants need that. So here when the electrons moves from photosystem 1 to primary acceptor ferredoxin, they don't reduce with the help of NADP reductase, they don't reduce NADPH, but they just return back to cytochrome complex to do chemiosmotic synthesis of ATP. So in response, ATP is going to form. So when enough ATP is formed, so the reaction is started to going normally when ATP is at sufficient amount. So this is mostly done in bacterial, except cyanobacteria. Lots of bacteria just perform this function, as well as what happened here, just ATP synthesized and no oxygen is released and no photolysis of water. So let's compare both of them, cyclic photophosphorylation and non-cyclic photophosphorylation. You see the difference in cyclic, it moves in the cycle like these electrons again come to photosystem 1 and then what is used, pyridoxine complex is used in that, cytochrome complex is used in that, and ATP and chemiosmosis is done. But here the normal process is going on. Here photolysis is done, oxygen is removed from here, and reduction of NADP also is going to be done. Cyclic and non-cyclic photophosphorylation, if we see some differences, only PS1 is involved in cyclic and PS1 and 2 are involved, both are involved in non-cyclic phosphorylation. Water is not required for cyclic photophosphorylation, and photolysis of water is done in non-cyclic. Oxygen is not involved in cyclic photophosphorylation, and oxygen is involved in photophosphorylation. Evolved actually. NADPH is not synthesized, and NADPH is synthesized in non-cyclic. So used to produce additional ATP in order to meet cell energy demands. So, but in non-cyclic, products can be used for light independent reactions directly. So dear students, I hope with this summary you understood all about the procedure of light reaction. And just keep this all the things in your mind, revise them. I hope you understood it. Wish you all the best. We'll see dark reaction in the next lecture. Allah Hafiz.